Because I said what was on my mind. I made a lot of enemies. I made a lot
Welcome, everyone, to Science from the Fringe. My name is Bryce Nickels. I'm a professor of genetics at Rutgers University, and I'm joined today by my good friend and colleague, Simon Wain-Hobson, who's a professor emeritus at the Pasteur Institute in Paris, best known for sequencing the first HIV genome and for his outspoken views on biosafety and dangerous gain-of-function research. Simon, how are you doing today?
Very well, thank you very much, and looking forward to discussing with you virology in every single nook and cranny that we care to go into.
So what we're going to do today, this is our first episode of what we are going to call In Defense of Virology. So Simon, do you want to explain what the title In Defense of Virology is about?
We cooked this up together, and basically it's not defending virology to outsiders. It's defending virology from the insiders. And as we've seen with the gain of function, the dangerous gain of function catastrophe, and the difficulty in handling the gain of the COVID origins question, people have very fixed agendas, and these people are virologists, and they're not giving the data and telling you the truth. And therefore, it's basically we're trying to protect virology from many of the virologists. Not all, of course, but there's just enough vocal virologists, and we have to sort of stand up and push back.
Many of the loudest voices are sort of drowning out the more sensible voices, I think.
Yeah,
I guess.
Flooding the zone, we know the expression, yes.
That's right. Flooding the zone with shit, I think, is the proper term, but I guess as an American, I can take more levity with the bad language. Right?
I will let you say that. I will stop for one or two words before.
So I did want to begin also by having you explain, talk a little bit about the essays that you've been doing over the past couple years that are published on the Biosafety Now Substack page. Could you talk about those?
Yes, of course. I mean, listeners, you should know that it's all your fault. Bryce and myself were in on a large conference call with 20 people or whatever, and I said something, and he said, "Simon, are you sure of that? Would you be ready to sort of, put that in writing?" And for me, of course, it was obvious that I had no questions about it. But to me, it was unusual to be confronted by this, and so we had a one-on-one call afterwards. And the chemistry worked, and I realized that this guy was just wanting to get to the data, no bullshit, get to the point, and find out what was there. And so he said, "Well, can I help you?" And we worked out this series of a thing called On Reading, and basically it's a weekly essay that's been going since January of 2024. And I think we're up to about 68 or 69 essays. They're weekly, apart from Christmas time and the summer holidays. I must let people some time to sort of breathe. And basically, I'm trying to sort of show how many papers in the virology literature are sort of warped. They're not telling you everything. There's some hidden agendas, and I basically try to, in maybe 2,000 words, keeping them small, just to sort of show that that some of the authors are just deviating. And so I always loved the song by Roberta Flack, "Killing Me Softly." And I'm trying to kill some of these papers in their own words and taking sentences and showing, do you realize that is a nonsense statement and that is contradicted by this, and simply doing it that way. So it's what scientists do all the time. They critique each other's papers. And so for someone like Bryce and myself, this is totally normal. but doing it for general readers, people with not a science background. So that is an art that I've learned, and I hope that my later paper essays are clearer, more focused than maybe the first 10, 15.
So let's get into the crux of the matter today, Simon, on In Defense of Virology, which is, let's define our terms. So
Yes.
what exactly is gain-of-function research, and what makes some forms of gain-of-function research dangerous?
We've had this semantic confusion, and then people have deliberately played on it. What we're concerned about is making new human viruses. Now, you can make human viruses from animal viruses, like bird flu. You can transform into a human to human transmissible viruses. This has been done as of 2012. That's what is the dangerous gain of function. This word gain of function has a historical sense that goes back 30, 40 years. None of that research is in question. It's this deliberate manufacture of new pathogens from animal pathogens, or the hyping or the revving up, the hotting up, if you wish, of an existing virus and making it nastier. So basically, it's making the world a more dangerous place. There's a little extra in to be added because, as many of your listeners will know, in 2005 we saw the resurrection, and that is the world of the 1918 Spanish flu virus. This is a virus that came, killed 50 million, moved on, replaced by other flu virus. It was dead as a dodo, like an extinct dinosaur. And then people found the genetic material in cadavers in permafrost, and they recovered the genes, and that was enough. But from that, they went one step more, and they made Spanish flu. So we now have brought back from the dead a virus which we know was highly pathogenic. It killed 50 million people. What more can you? 50 million is more than COVID. Remember that back in 1918, the world's population was 1.8 billion, and now it's 8.2. So yeah, this is being done. And so basically, these are the dangerous areas. Everything else we can push to the side. They are the vast majority of virology research is responsible. It's done, but this was a step too far, and it occurred with our increasing technology. But it came to the fore with NIH-sponsored research in 2012.
Can you talk about what the main dangers are of gain of function, or the dangers of the risky gain of function research?
Yes, just go back to that 2012 period. There were a couple of papers where they had transformed bird flu virus, something with a name called H5N1. This is the one that is mutating, going around. Even today, this is the one that infected cows in the USA. And they made sure that this virus, which is not transmissible between humans, it can infect humans, and it can kill 60% of humans infected, but it doesn't transmit between humans. So you can say, ooh. And what did they do? They made it transmissible between ferrets. And people in the flu community have this rule of thumb: if it's transmissible through aerosol route between ferrets, it's probably transmissible between humans. So they made a new human flu virus. Your readers or listeners may not know, but there's no one in the world who has natural immunity to H5N1. So if this virus was to come in and start transmitting and generate even an epidemic, it would be pretty nasty because we don't have pre-existing immunity. Like your antibodies to last year's seasonal flu wouldn't do a thing. So this is being done, and it's being done unquestioned. And there are few people in the world, like yourself and myself, who want to sort of say, wait a minute, there's, NIH, you have a problem.
So what did they claim the utility of those experiments would be?
They said that we could predict the next pandemic virus. We could anticipate the next pandemic virus. And if we could do that, which they said we could do, when we could make drugs and we could make preventive vaccines, put them in the freezer, wait, and when it came out, we would just multiply everything by 10,000 and we would nip this pandemic in the bud. This was the logic of 2012, and as everyone will remember, December 2019 came and January 2020, and we had something called COVID, which is a coronavirus, totally different virus, and it wasn't this H5N1. And in fact, it's rather frightening to say, but one of the protagonists of that work back in 2012 has now written that H5N1 is not a threat for humans. So there was lots of hype, lots of drum beating, and with the long run, because you and I, we know in science we have a look back period of 5, 10, 15 years sometimes. It didn't pan out, but we were sold this pipe dream and told to suck it up.
Was there any utility of the results? Like, how would you quantify the benefit?
Yeah, I get you. So for one, no one could put a number. You can only put words like may, maybe, perhaps, could, possibly, which I think everyone will agree is anything but precise science. Science is about numbers, quantification, and something say like maybe. Well, that doesn't help much. The other answer is was there anything there for public health officials? No, zero. I've given you a number, zero.
Zero. I would agree. So definitely not a risk worth taking, I guess, to paraphrase the famous headline of the op-ed written by Fauci and Collins and Gary Nabel back in 2011.
In the Washington Post. This is an op-ed, so it's an opinion. So you can say these gentlemen had, the right to free speech. The problem is when the right to free, this is speech coming from 3 of the top people in the NIH. So first, they gave an opinion in public when none of the scientists, like the virologist like myself, he'd even seen these papers, the manuscripts, because they were not published. They gave an opinion saying it was okay, and no one was allowed to dissent. So they had their right to free speech, but because of their position, they stifled everyone else. They squashed everyone else's right to free speech. And science is bottom up. It's not top down. So things from top down in science always goes wrong. We know that there are top down situations. You can't have a company and have there's a certain degree of top down, but science is bottom up. No one knows where the next discovery is going to come from. No one can tell you. You and I can't say, we know who's going to be the next Nobel in 20 years. It's absolute nonsense. We don't know because the discoveries come from the bottom, from unexpected places. So we were squashed. And it turns out, as I said, Karaoke has said that there's nothing in this and it's not a threat to humans. So these people were just left, and that op-ed is left shanghaied, high and dry. You choose the phrase you want. They got it wrong because history and time shows they got it wrong. So science should be left to the people doing it, and if it takes time, it takes time. And this was a category error. This op-ed should not have. It was a faux pas. How can I say it?
So this is an op-ed written in December of 2011,
Exactly.
at the time when the controversy around these experiments was emerging, and there were a lot of
emerging, but no one had seen the documents apart from the insiders, so no one could comment on the quality, yay or nay. Whether you agreed or you disagreed, you couldn't even agree or disagree because no one had seen it. It was all whatever, and we were told in an op-ed. But when you have the representing the biggest funders of biological and infectious disease research, if Wave said this, everyone's going to bow down and sort of say, okay, I believe you. And that's exactly what happened. No virologist questioned it. There's only one professor of virology in the world who has said anything, And he's speaking to you now. One. There were other people who were not virologists, very brave people who were epidemiologists and biochemists who knew enough to realize this was nonsense. But all the virologists just kept mum, shut up. It was an omertà, a global omertà, which is stunning.
And you think the op-ed and Fauci and Collins staking their position on this was a big driver of stifling debate in the virology community?
Oh yeah, but for me, it's obvious. It doesn't need to be. And on top of that, you never should prejudge. You know, you listeners will know. Bryce and I, we've been in this for quite a while, and we know how difficult it is to see into the future. Sometimes we can't see more than 6 months, 12 months, and then we get something. Say, God, how did we miss this? We must have been. What were we thinking of? But doing science is tough, and looking into the future, we get it from time to time. It's hard, and we should go by the data. And there was nothing in this gain of function, this dangerous gain of function for any public health official. And so when COVID came along, there was nothing in there, and there's nothing in there for making vaccines. No, it was an aberration, a delusion.
They create these souped up viruses in the lab, and those present a risk to public safety if they happen to get out through infecting a lab worker.
Correct.
What about the informational risks? Can you talk
Ah.
about how not just the creation of a new virus presents a risk, but the information creates a potential threat?
Yes. So the technology in genetics, what we call molecular genetics, is absolutely phenomenal. When you and I started, well, you're a younger man, but when I started, I mean, we know how to sin a gene, and the virus getting a leaky virus would be the issue. But today, we can take the genetic sequence from a database. I mean, you could go to the database and pull down a sequence, and we can then chemically synthesize the nucleic acid and make a virus from that. These techniques are out there, are available on the web. It's done, and it's done for most viruses. Perhaps not all human viruses, but 90% plus. Some viruses proved a little bit difficult. So what we call the DNA viruses are relatively easy, like HIV, retroviruses. And then came what was called reverse genetics, where you're copying RNA back into DNA. This is jargon for us, but we can do it. So you can, from a simple sequence, you can derive the gene, you can chemically synthesize it, and from that you can make the virus. So if I sent you, or you sent me a sequence of some novel virus from South America, say, let's call it, it's Andes virus. It's an interesting virus in there, which is not infects a few people, but it can transmit from humans to humans, unlike H5N1. So we should really keep our tabs on this one. So if you gave me that sequence, says, Simon, you're a virologist, could you make this virus in the lab? And you said, I would like to have it, and could you do it for me because we're friends? So I said, sure. It could be done. And I'm now retired, but my lab could, if you gave me that sequence, we could have make virus within 1 month. So the fact is that as soon as this is available, published on the web, courtesy of the US government, paid for by the US taxpayer, this manuscript can be downloaded in Iran, North Korea, Russia, Cuba, wherever you want. They can just make the gene. They can recover the virus. It's not rocket science. It's not anything like as complicated as purifying uranium to get 96% enrichment. It's much easier than that. Hundreds to thousands of labs across the globe could do it if they put their mind to it. So that information, and you're absolutely right, that information alone is enough to let someone who is misdirected, hates the West, or is just bloody minded and say, well, I want to do it. And there it is. And this discussion has not taken place. We've discussed the gain of function amongst virologists and whatever, and people talk about how can we improve, whatever. But this thing about just pushing the information out there is not discussed. And I think we have to do it. We can't avoid it, but there's great resistance to do it because people want to do the nice things. They want all the fun and not the responsibility involved. But this gets back to human nature, so I'm not going to surprise you.
Well, there has been discussion about trying to do some sort of regulation around ordering of DNA sequences through companies.
Yes.
And that would get at some of the threat of synthetic viruses, wouldn't it?
Yes, I mean, Bryce is he's referring to the fact that before making the virus, you have to do the nuts and bolts of synthesizing the gene precisely from the information. And so computer programs allow you, then you can order it. And so if you're some sort of nut or whatever, and you're asking for this, or you have one of these sequences, the mutation in which you feel will make the virus even nastier. So people are asking the companies that do the chemical synthesis to screen the people who are asking and ordering, and do a prior screen, a security screen. And if something goes up, they will probably then filter it through to the FBI, and the FBI will sort of just take a look and maybe drop by, which is all very fine. But what can the FBI do to a lab in Russia, Iran, North Korea? Peanuts, nothing. No, of course not, nothing. They have the money, they have the facilities, they have the technology to do it. So this screening process, which looks and sounds very great, will make life harder for the loner to do it. But as soon as you've got a little bit of muscle power behind you, or financial power, if you want to go off grid, you just buy a DNA synthesizer, hire a technician with experience, and then you can make as many things as you want under the radar, and all these well-meaning screening programs will just fall flat. So yes, it will stop the loner, maybe the crazy, the garage scientist, as I think one of the expressions, but we don't know who the threat is going to come from.
Is there a difference at all between dangerous gain of function research and bioweapons research or bioweapons development?
Let's go back straight to those 2012 papers on bird flu. For me, there's not a single difference between that coming from a civilian lab in renowned, publicly funded universities, things like University of Wisconsin, Rotterdam, Harvard, you name it, Mount Sinai, wherever you want, and a military lab somewhere making the same thing. It's indistinguishable. The result, you can say, well, the intent, these gentlemen who made it didn't intend to make a biological weapon. Yeah, sure, I agree. And whereas the military intend to make a biological weapon, if only to threaten, not necessarily to use, but to threaten. But the result is the same. But there is a big difference. The civilians publish their genetic blueprints on the web, and we just been to this, where you said, well, the information is enough. When you see a sequence as a geneticist, you can say, I can make that sequence. I can pick it off a piece of paper and I can remake that thing and get that gene. Whereas a military people, everything is hush-hush. All you have to know is that you have this threat. It might be bullshit. It might just be hubris. It might just be fake news. But if you say it with enough panache, people might take you seriously. Whereas these people are actually making and publishing their blueprints free of charge for anyone to get. So the civilian approach is indistinguishable, and they commit the ultimate error of letting their adversaries into the secret and allowing them to do it for themselves. It's total madness. It's unthought of. And I've asked a few people who know about the biological weapons, and they said, no, everyone keeps the secrets to themselves. It's the threat, it's the fear of. Whereas these people are saying, here it is, this is the fear, I've got it. And they say, they don't say, well, I'm not making a biological weapon. But someone could take your information and do it. So it's a height of irresponsibility. It's exactly the same. It's exactly the same. You're right. You're absolutely right.
Perhaps for clarification, is it simply that in order for something to be a biological weapon, there has to be intent? Is that the only reason, like intent to use it as such? Is that the way in which we're parsing the terminology between something that could be used as a weapon and something that is a biological weapon? Is that the person making it has to have the intention of using it?
I should probably speak with more people, because I've always been from the other side, trying to sort of beat the daylights out of microbes and make the world a safer place. My wife's a pediatrician. My son's a cardiologist. The thing is to attack and beat the daylight of the disease and whatever. And then intent, I suppose it is intent. I mean, I actually don't know what goes on in the head of people doing such work. But if it's to threaten, and therefore to make your adversaries think twice, then yes, intent, certainly intent. But the intent maybe just from myself, and our intent is to beat the hell out of microbes. And I know from discussions with we have exactly the same position. I know. From Pasteur Koch through to yellow fever, to malaria, to the guy who got, I'm blanking on his name, polio virus, and the Hep B and then Hep C, beating these things, beating the daylights, and then you get the biotech coming in and pharma making drugs and killing HCV. Do you know the wonder of HCV is that HCV causes cirrhosis of the liver, and I was listening to some liver doctors, and they were saying, cirrhosis is reversible. They never knew that, and through drugs for Hep C, cirrhosis is a reversible process. Fantastic. And you can cure some people with these drugs. Unbelievable. And there we are. We have a few people, not many. Listeners shouldn't be sort of flippant, sort of saying, all you bloody virologists are doing it. No, but there are just one or two, and one or two too many. And unfortunately, there hasn't been pushback. In the US, there's been discussion, but in Europe, there's been no discussion. And outside, a little bit in Germany, a little bit in the UK, Switzerland, nothing. The Scandinavian countries, nothing. And the rest of the world, no. It's how can I say, it's the whole community is in denial, totally in denial.
A lot of people will argue that it's too difficult to define what dangerous gain of function is. I'm sort of smiling at this because I think this is absurd. But what do you say to people who say it's too difficult to define what dangerous gain of function is, and this is a very difficult subject to deal with because it's hard to identify things? What do you say to them?
So I'll answer the question directly and then back up. This represents an infinitesimal fraction of research into bacteria, parasites, malaria, viruses, and whatever. Absolutely trivial. Not, it's not like 0.001%. It's absolutely so hardly worth speaking about. And yet it has captured the imagination because it's so dangerous. So it is a very minor form. So your listeners can be reassured, it's not the whole of virology has gone crazy. There's just one or two sort of irresponsible thoughts, and we've got to contain this. But, if things aren't contained, then other people will sort of say, well, maybe I can do that too, or they might feel encouraged to do it. Why I I I think it's a poor misunderstanding of science. And you mentioned language, and unfortunately, we have seen uh, virologists deliberately muddy the waters. And if ever you need precision in words, you need it in science. Okay. Poets can use and play on words, and comedians can make puns and have used the ambiguity of language and make us all laugh. But scientists should have to be as precise as hell to make sure that you get your terms right and that I'm not confused. And they have been wishy-washy, ambiguous to the extent that it's part of, it's not a cover-up, but they just don't want the topic discussed. I wouldn't want to say why this is. I'm sure they're embarrassed by it, but I actually don't know. I don't want to get inside their heads because the science and the logic behind it is wrong, and therefore I'm not going to get into. I don't want to get sidetracked into understanding why is someone wrong. I can say why they're wrong. That's it, and what they think and why they think that. Well, I don't have time for that. There's enough to do. Some of us have been fighting this since 2012. That's what, you can see my grey hair. That's what, 13 years, and it's not finished. So we've got to concentrate on the big stuff. I don't know why there's been such support for it.
Perhaps that's where we could conclude this first episode of In Defense of Virology. So we have begun the discussion of talking about what gain of function is, why it's dangerous, not just from a perspective of lab accidents, but also from providing information to adversaries that could use that against us. I guess the other thing, maybe you want to end with the importance of defending the field of virology from virologists that are performing dangerous gain of function research and also defending it as something that's valuable to public health. Do you want to say something about that, Simon, before we wrap up this episode?
Yeah, sure. I wrote one of my essays is why virology is the queen of the biological sciences. Virology, I mean, I could speak for hours. It's it's a wonderful subject. It's so complex. It's so subtle. There's so much to learn about biology and disease. And of course, as a virologist, you're in this game where you can actually do some good and help your fellow citizen beating the hell out of microbes. I'm not in love with viruses, but I admire the subtlety. It's a fabulous subject, and sometimes I say, I can't imagine why you'd want to do anything but virology. But um, um, where were we? I got carried away with myself.
Well, you're talking about why you love the science of virology, studying viruses. And
Yes.
perhaps to maybe put a finer point on this, we're in a situation now where many people in the public are very distrustful of
Oh,
scientists and are in a and
yeah,
coming on the heels of a pandemic that was caused by virology. How do you get people in the public to say, wait, we want to continue giving money to virology?
yes. Well, very, very simple. I mean, something called smallpox eliminated by vaccination. Your listeners will not know that there's a measles-like virus of cattle called rinderpest, which caused huge economic losses in cattle, eliminated by vaccination. Polio 2, polio 3 gone. Polio 1 on its last legs. And polio 1 goes back to Roosevelt and the history of that. And virology helps us to do good. We can make drugs. So HIV, no vaccine, but it's a manageable disease if you can have access to the drugs. Papillomaviruses that to cause cervical cancer in women, no drugs, but an incredibly efficient vaccine. And all this came out of basic virology. So I would say, as usual in anything human, some people get us into a mess, others get you out of that mess. And we got into this mess through a handful of people The others must help us get out of it. So we need those virologists to get out. Also, training a virologist, it's not something you can just do quickly. There's a lots of experience. As I said, my son's a cardiologist. He's interventional cardiology. That's 15 years training. You just can't sack all air traffic controllers or all cardiologists and sort of say, we'll get a new generation. You can't do that. So I think this is noise that has happened. Unfortunately, got virology a very bad name. Virology needs to be defended. So in defense of virology, and it's going to be some of the virologists and who will sort of set the clock, and people will realize, yeah, we need it. And on top of that, as I say, it's just such a fabulous subject for learning biology and its impact, its knock-on effect of virology on oncology, on vaccination, on diagnostics. I mean, when HIV broke, it transformed virology and immunology. And virology and immunology became modern. And the world is a better place for it. So it has tremendous and immediate effects. Sometimes it takes 5, 10, 15 years, but sometimes, do you realize your listeners, they probably had a PCR test in a tent on a street corner in some city? You're doing virology on a street corner. It's absolutely outrageous. It's wonderful. And it shows you what we can do. And this sort of comes, and we can help you. Yes. So if you're positive, stay at home. Don't infect other people. Keep this damn virus under control and whatever. And when you're out, you go out. For me, it's a triumph for virology. But as you said, the origins of the virus, did it escape? And we, from a strictly scientific point of view, with hardcore, where's the data? And the data is not forthcoming, but it doesn't look good. And all my virologist colleagues who tried to make the pitch for a natural, their cases, I understand the arguments, but it's a weak case. For me, if I was a judge, I wouldn't give them the trial. I would throw the trial out. Yep.
Well, you're too kind on that. I would have less measured words about that if I were going to say anything. But I think I've said a lot on Twitter and other places about that already.
No,
but,
you can make a good circumstantial case that might work in law, but for science, science is yes, no. It's not a question of opinion. It has to work. So, for example, just take a smartphone. You can't make a smartphone out of beliefs. It has to work, and you have to be reproducible, and you have to check it, and you have to do it. And if it doesn't work and you can't reproduce it, it's crap. You can't make a smartphone out of beliefs. So that's the only difference. So, but it doesn't look good. I agree, absolutely, it doesn't look good. And the gain of function. My colleagues made this thing. They did a disservice to virology. For me, unquestionable disservice to virology. Unquestionable.
That's the goal here, where instead of, I guess the point is that what we're going to try to do is...
And we're going to push back, and we're going to defend virology from the virologists. So we've gone the whole circle, and we come up, and we can go our own ways and come back for another one.
What I'm going to do now is I'm going to sign off on the first episode of In Defense of Virology. Thanks, everyone, for joining us. And I would recommend that everyone check out Simon's essays over on the Biosafety Now Substack. And also, please, I guess I need to end these, Simon, by saying, please like and subscribe to Science from the Fringe. It sounds like a real...
Subscribe, and as I said, it's all Bryce's fault. He's behind. Yeah. So do it for Bryce, please.
Do it right. Do it. Well, right. Do it. Because, just do it. How about that? Because we need something. I don't know. It helps. If they subscribe, it increases the algorithmic, the calculation that the computers use to spread it across the globe. And we need reach, Simon. We need these to reach the globe.
Yes.
So that's what we're trying to do.
But our number one goal is to make sure that I don't like the word truth, but to make sure that the reality gets out there and people who are looking to sort of say, well, what's the reality? I say I don't like the word truth because it's a loaded word that has meanings from 2,000-3,000 years. But the reality, where's that? And hopefully they can come across you and me and sort of get something, and we don't ram it down their throat and say, here it is, here's some references, and look at it. And if you don't agree, if there's something that's not right, post something, and we can sort of pick it up and see. But the idea is to help people understand and filter through this massive disinformation, which unfortunately virologists have subjected people to. Yes, so again,
the disinformation from the virologists, I think, to make it clear what we're talking about, because there's enough people. Yeah.
In Defense of Virology is working on multiple planes, and we're trying to help the non-virologists sort of get to grips with this. And something has gone wrong, and we're going to try and sort it out.
Okay. And with that, I'd like to thank Simon once again. And until next time, this has been Bryce Nickels with Simon Wain-Hobson on Science from the Fringe's In Defense of Virology. Thanks, everyone.
Thank you, Bryce.
What is she?